Hello, everyone, and welcome to our module on cells of the nervous system. So, when you think about cells of the nervous system, most people think of neurons, and those are definitely very important cells of the nervous system, and we'll talk about these in this video. But there are also all these cells that support the neurons, which I've listed here, and these are also important to understand. These cells can be affected by disease states, they can turn into tumors. So, in this video, we're going to go through the neurons, but we're also going to go through all these supporting cell types as well. Some of the cell types we'll discuss in this video are found in the central nervous system and some are found in the peripheral nervous system. So let's talk about those two different nervous systems. So basically, when we say the central nervous system, we're talking about the brain and the spinal cord. But then, as you may know, nerves can leave the brain or the spinal cord and travel outside of those parts of the body. And when they do this, we call them parts of the peripheral nervous system. So, for example, the cranial nerves, which exit the brainstem and innervate muscles in the head and neck. Those are parts of the peripheral nervous system. So are spinal nerves, and so are other peripheral nervous system nerves. Now, before we get into talking about specific cells, let me talk about a term you'll hear a lot when discussing the nervous system, and that is glial cells. So the word glial means glue, and so these cells were called glial cells because they were thought to be the glue that held together neurons. And glial cells are all the cells that support neurons. And we broadly divide them into two groups. The macroglial cells are larger. They're about 10 to 20 microns in size, and this includes astrocytes, oligodendrocytes, and ependymal cells, and we'll talk about all of these in just a minute. And then there are the microglia, which are much smaller cells, 5 to 10 micrometers, and this basically means the CNS macrophages. The CNS macrophages can also be called microglial cells. So related to the term glial cells, you'll sometimes hear the term gliosis, and this basically means proliferation or hypertrophy of glial cells. And this is what happens anytime there's injury to the central nervous system. You will see gliosis, and that is the central nervous system attempting to heal itself and regenerate tissue. And the key cells for this process are astrocytes. It's high yield to know this. We'll talk more about astrocytes in a minute, but anytime there's injury, astrocytes become activated, and they're very important for determining the healing and recovery from a central nervous system injury. You also hear the term glioma. This refers to a tumor that arises from some type of glial cells. There are many types of gliomas, including astrocytomas, oligodendrogliomas, and ependymomas. Okay, so now we'll start discussing the individual nerve cells that you can find in the nervous system, and we'll start by talking about the neurons. So this is a depiction of what a neuron looks like on the slide here. It's got a large cell body, which is what's shown here, and the cell body is also called the soma of a neuron. And in the cell body, you can find the nucleus. Coming off of the soma or the cell body, you have little fingers that are poking out. These are called dendrites, and this allows the neuron to communicate with other neurons. Then you have this long nerve fiber here. That's called the axon of the neuron. And at the end of that fiber, you have presynaptic terminals. These are regions of the axon that can release neurotransmitters. Now, along the length of the axon, you find structures surrounding the axon called myelin or myelin sheaths. And these are very important because they improve the ability of the axon to conduct signals. And we'll talk more about how myelin does this in a minute. There are cells that produce the myelin sheath, and in this example, they're showing a Schwann cell, which is one of the cell types that can produce myelin sheaths. Schwann cells do this in the peripheral nervous system. Other cells do it in the central nervous system, and we'll talk more about that in a second. And then there are these little spaces of the axon not covered by myelin. Those are called the nodes of Ranvier. And so the reason that myelin is so effective for allowing signals to be conducted is because signals can basically jump from one node of Ranvier to the next. So instead of an electrical signal having to traverse this entire long axon, it can basically jump from one node of Ranvier to the next. And that's what myelin does, is allow this to happen, and this speeds up conduction of impulses along the neuron. So you can think of neurons like electrical fibers. They basically conduct electricity. And they do this by allowing positively or negatively charged substances like potassium and sodium to move in and out of the neuron. So to understand this, let's imagine that we've got a length of an axon right here. Well, at rest, there are relatively more negative charges inside the axon and relatively more positive charges on the outside of the axon. This creates what's called an action potential, meaning a voltage across the membrane of the axon. And at rest, when a neuron is not conducting an electrical signal, that resting potential is negative 70 millivolts, and that's what's shown right here in this image. Now, if this neuron wants to transmit an electrical signal, it's got to allow positively charged ions to come into the cell, and this positively charged ion movement will travel down the length of the axon, and that's how signals are transmitted along those axons. The way this happens is depicted in this image on the slide here. So what happens is a signal comes in and tells the neuron to depolarize right here. That's this stimulus. And if the stimulus is weak, you will have a failed initiation of an action potential. But if the stimulus is strong enough, it will allow sodium ions to come inside the neuron. Sodium ions are positively charged, so when they start to come inside, the voltage starts to go up from -70. And it goes all the way up to about +40. But what happens when it reaches about +40 is all the sodium channels are open. As much sodium as can come inside the neuron has already occurred. And now what's going to happen is potassium ions are going to start to leak out of the neuron. Potassium ions are positively charged, so when they leave the neuron, that is going to result in the action potential coming down. And that's what creates this phase right here, repolarization. What you have is the charge across the membrane, the action potential comes downward and it gets very, very negative, and it comes back up to that resting state of -70. And so the key things to know about the way the voltage changes across the cell membrane of a neuron, which is the action potential shown here, is that first of all, some stimulus has to come in right here to get the process going. Often that stimulus is from another neuron that tells a second neuron to depolarize, but there are other stimuli that can get this process going. What that's going to do is allow sodium ions to come in. That's responsible for this upward trajectory here of the action potential. And then in repolarization, you're going to have potassium ions leaking out, and that's going to cause the membrane voltage to go back down. And then it's going to briefly hyperpolarize, but then come back to the resting state of -70 millivolts right here. And as you can see in the x-axis here, this whole process just takes a few milliseconds to occur. So this is a very, very rapid process going on all the time in the neurons inside our brain and in our peripheral nervous system. So on this slide, what I've done is I've listed the key facts you want to remember about neuron action potential. So first of all, you should be aware that at rest, neurons have a voltage of -70 millivolts. And I didn't talk about this on the last slide, but the reason that the voltage is maintained at -70 is because there's always a leak of potassium ions out of the cell. This is going to make the charge across the membrane negative, and that's what gives you that -70 millivolt resting potential at rest. To depolarize, sodium channels have to open. This allows sodium into the cell, and that's what raises the voltage. And then what's going to happen is, as some sodium channels open in the axon, they are going to open other sodium channels further along the axon. In other words, the depolarization is going to propagate. So imagine that you have a long axon like what I've shown here, and we get some sodium coming in down here. What that's going to do is trigger the opening of sodium channels here, so sodium can come in here, and that will trigger some more opening of sodium channels. And you're going to get positively charged ions coming in and moving down the axon like this. And that's how these electrical signals of neurons are propagated. At the end of the axon, at the terminal, what usually happens is calcium channels open, and that triggers the release of neurotransmitter. I talk about this in a lot more detail in other videos. Usually there are neurotransmitters stored at the end of the axon inside small vesicles. These are small membrane-bound structures. And when the calcium comes in, the vesicles fuse with the membrane and they release their neurotransmitters. This is called exocytosis. And so this whole process that goes on at the end of the axon is depicted right here. What happens is these voltage-gated calcium channels open when the nerve impulse reaches the end of the axon. That causes these vesicles to fuse and release their neurotransmitters into a synaptic cleft. And then those neurotransmitters can go and exert various effects on the other side of the synapse. This could be to activate another neuron. This could be to tell a muscle fiber to contract. There are lots of different effects that neurons are designed to cause. Okay, so all that stuff about action potentials in neurons is great, but what's the clinical relevance? Well, first of all, you should be aware that agents that block sodium channels can inhibit neuron signals. This is the way local anesthetics work. I have a whole video on local anesthetics in the anesthesia section, but they include all these drugs right here. The way they work is they block sodium channels, and this makes it so that nerves can't depolarize and can't transmit signals. And one of the signals they can't transmit is sensory signals about pain. That's why these drugs work as local anesthetics. There are also some neurotoxins found in nature that block sodium channels. For example, there's a fish called a pufferfish, which produces tetrodotoxin. This can block sodium channels, and this can actually cause paralysis because you can't get depolarization of motor neurons that tell muscles to work. Okay, now I'm going to move away from neurons and talk about other cell types of the nervous system. Some of these cell types are found in the white matter, and some of these cell types are found in the gray matter of the central nervous system. So let's talk about what I mean when I say white matter and gray matter. So white matter basically is created in the brain by myelinated axons. So let's look at this little slice of the spinal cord right here. You can see that there's a gray part in the middle, and this is what you see anytime you look at a cross-section of a spinal cord. You see the gray part in the middle, but then there's all this white out here. So the white part is the white matter, and that's got all the axons running in different directions carrying signals. It also has some glial cells like oligodendrocytes, but mostly white matter is made up of myelinated axons. So you can find this around the gray matter in the spinal cord, like in this image right here. You can also find it under the gray matter in the brain. That's what's shown right here. We've got gray matter on the outside and this white matter here in the middle of all those myelinated axons running in different directions. The gray matter, on the other hand, is mostly made up of neuronal cell bodies. So that's what makes the center of the spinal cord image right here look gray. It's all those cell bodies. It's also made up to some degree of the dendrites attached to the cell bodies that we saw earlier and the axon terminals and some glial cells. But mostly, when I think of gray matter, I think of cell bodies of neurons in contrast to the axons, which make up the white matter. And so you can find gray matter also in lots of different places in the central nervous system. You can find it in the cerebral cortex, the deep nuclei, which are part of the central nervous system, and also in the central spinal cord, just like shown in this image right here. Okay, so now that we've gone through that, let's talk about some more cells of the nervous system, and we'll start by talking about astrocytes. So astrocytes are cells that support neurons. They're found in the central nervous system. You can find them in the gray matter and also in the white matter. They have a number of important roles. One of the things they can do is remove excess neurotransmitter. But beyond this, one of their most important roles that you want to be aware of, especially for the Step 1 exam, is that astrocytes are responsible for repair and scar formation when there is brain injury. Like I told you before, anytime there is injury to the central nervous system, a process begins called reactive gliosis. This means hypertrophy and hyperplasia, and it's mostly of astrocytes. Those are the cells that are going to go and try to repair the damage to the central nervous system. One of the major markers clinically of astrocytes is GFAP. GFAP stands for glial fibrillary acidic protein, and there are a number of ways you can test for the presence of GFAP, and when it is found, that usually means you're looking at an astrocyte. So this is important for identifying tumors that come from astrocytes and just in general, identifying astrocytes in the central nervous system. Let's talk about some clinical relevance around astrocytes. So first of all, there's a tumor that I talk about in another video called an astrocytoma. It usually develops in the cerebellum in children, and it's going to be positive for GFAP because, as I told you, GFAP is a marker of astrocytes. And then super high yield to know is that there is a virus called the JC virus, which can infect astrocytes and oligodendrocytes in the central nervous system. And in patients who are severely immunocompromised, this virus can cause a central nervous system disease called PML, which is progressive multifocal leukoencephalopathy. It's a very rare central nervous system disease that mostly occurs in advanced HIV disease when patients are severely immunocompromised. It presents with general symptoms of central nervous system dysfunction, things like confusion, motor loss, and seizures. But the thing you want to remember is that this is caused by the JC virus, and the cells that the JC virus likes to infect are astrocytes and also oligodendrocytes, which we'll talk about in a minute. And this is an image of a patient with PML on the slide here. These white areas in the brain are evidence of PML. You don't have to know how to diagnose PML by MRI or CT of the brain. I'm just showing you this to remind you of the fact that this virus infects these cells in the central nervous system. Okay, moving on, our next cell type of the nervous system we'll discuss are the microglia. And as I told you earlier in this video, microglia is just another name for macrophages that reside within the central nervous system. And these macrophages, just like other macrophages, can proliferate in response to injury. After injury to the central nervous system, they can become larger phagocytes that consume debris and other things like that. Most important thing to know about microglia in terms of the Step 1 exam is that HIV can persist in the brain inside these macrophages. This is something that's been studied extensively in the basic science literature. When you treat patients with HIV with antiretroviral therapy, it can be difficult to completely eradicate the virus from the body. And one of the reasons for that is because the virus likes to hide inside of central nervous system macrophages. Along these same lines, in patients who have chronic HIV and develop encephalitis, you can find nodules of activated microglia that are being triggered and are growing in response to the HIV virus. So always think of these cells as associated with HIV. Okay, our next cell we'll discuss are the Schwann cells. These are cells that produce myelin for peripheral axons, not for the axons in the central nervous system. That's done by a different cell type, which we'll talk about in a minute. Each Schwann cell myelinates one single axon. This is depicted here in this image. So you've got a Schwann cell, and it's producing myelin for this single axon right here. And in another video, I talk about the response of peripheral nerves to damage. And you should be aware that the Schwann cells are very important for neuron regeneration after peripheral nerve damage. And you can imagine that just by looking at this image here. If we were to have some type of injury to this nerve, whether or not the Schwann cells survive and can produce more myelin will be a factor in whether or not the axon can regenerate. You should also know that Schwann cells are destroyed in the Guillain-Barré syndrome. I talk about this in other videos, but this is a rare syndrome often triggered by infection or other things that leads to loss of peripheral nerve function. This can lead to sensory and motor problems. And then also in another video, I talk about tumors of the nervous system. Schwann cells can form schwannomas. These are also called acoustic neuromas. They classically affect cranial nerve 8, and I talk about these in detail in another video. Okay, our next cells we'll discuss are the oligodendroglia, also called oligodendrocytes. And these are the cells that myelinate central nervous system axons. So they are the counterpart to the Schwann cells in the peripheral nervous system. One thing that's different about these cells than Schwann cells is that each oligodendrocyte can myelinate multiple axons, not just one, like we saw in the last slide. So this is depicted here. You've got an oligodendrocyte, and it's producing myelin sheaths for multiple different axons. And these are the most common glial cell found in the white matter, and hopefully that makes sense to you. The white matter is made up of axons, and so you need to have a lot of oligodendrocytes around to create myelin for those axons. In terms of pathology, what you want to know about these cells is that they're destroyed in multiple sclerosis. Multiple sclerosis is an autoimmune disease that I talk about in other videos. It's the classic example of a demyelinating disease, and it leads to a loss of nerve function without myelin. And the reason this disease happens is because you lose function of oligodendrocytes, and therefore you lose myelination of central nervous system axons. This can present with clinical features like weakness and sensory loss. There's a classic presentation I talk about in the ophthalmology section, which is optic neuritis, where patients present with painful visual loss. This is from loss of myelin in optic neurons. Patients can also present with problems moving their eyes laterally. This is called an intranuclear ophthalmoplegia. And I talk about all these things in other videos. And then finally, another thing to remember about oligodendroglia is that they can be infected by the JC virus, which I mentioned earlier. This can lead to PML in patients who have advanced HIV. The two cell types that the JC virus likes to infect are the oligodendrocytes and the astrocytes. Okay, let's talk briefly about myelin itself. So myelin is a substance made up of lipids and proteins. And as I mentioned earlier, the purpose of myelin along axons is to increase the speed of impulse propagation. Because of myelin, neurons can send signals via what's known as saltatory conduction. The word saltatory means to leap. And so this type of conduction means that signals can leap and go from one node of Ranvier to the next. This is what I mentioned earlier. So if we look at this drawing over here, the purple things here are myelin sheaths. And so instead of having to depolarize this entire neuron like this, all that has to happen is you have to depolarize this little node of Ranvier here, and then you can jump and depolarize this one here. This makes the entire process of signal propagation faster. It increases the conduction velocity. It also increases another metric known as the length constant. This is how far a signal can travel before it peters out. And as we've already discussed, in the central nervous system, the cells that produce myelin are the oligodendrocytes. And in the peripheral nervous system and peripheral axons, the Schwann cells produce myelin. Okay, let's talk about the different types of axons or nerve fibers you can find in their body. Some are large, some are small, some are myelinated, some are not. And each of these different axons serves different purposes. So we categorize nerve fibers by whether they are large or small, and also by the presence or absence of myelin. And when I say large or small, I'm talking about the diameter of the axon. So the biggest nerve fibers are the A alpha fibers. These are large myelinated fibers. They can be up to 20 microns in diameter. And this is what you find for most of our motor fibers. Fibers that are going to innervate muscles to contract, those are usually A alpha fibers. You also see A alpha fibers for some forms of sensation, things like touch, vibration, and position or proprioception. The next size down are the A beta fibers. These are also large and myelinated, but they're a little bit smaller. They're up to 12 microns in size. And these are fibers that transmit signals for touch and pressure. Then we've got the A delta fibers. These are small, up to 5 microns in size, but they are myelinated just like all the A fibers. And these carry signals like cold and pain. And then finally, C fibers are very small fibers, up to 2 microns in size, and they are unmyelinated. And these carry two sensory types of signals, warmth and also pain. And so it's kind of annoying, but on your USMLE exams, they are going to describe various nerve fibers that transmit certain sensory functions or maybe motor fibers, and they're going to want you to know how big or small, and whether the fibers are myelinated or not. Okay, the last thing we'll talk about in this video are structures found on neurons that allow us to sense the world around us. So there are 4 structures you can find on nerve endings that allow us to sense the world. Those 4 structures are shown here. Each of them senses different things, and that's what we're going to talk about in the next few slides. Okay, let's start by talking about free nerve endings. These are mostly found in our skin, and they can sense pain and also temperature. And there are different receptors on these free nerve endings that detect pain, cold temperature, and warm temperature. And so for this reason, there are different nerve fiber types associated with free nerve endings. As we talked about a few slides ago, the C fibers are what send information back to the brain about warmth, and the A delta fibers are what send information back to the brain about pain and cold. Meissner's corpuscles are touch receptors. They're located near the surface of the skin. They're concentrated in surface skin tissue, like the tissue that covers our fingers. The skin that covers our fingers is called glabrous or hairless skin, and it's a specialized region of skin that has a high concentration of Meissner's corpuscles, so we can sense fine touch. The way these receptors work is they're deformed by pressure, and this leads to nerve stimulation and signals that go back to the brain. And as we talked about a few slides ago, it's those large myelinated fibers, like the A alpha and the A beta, that send touch signals back to the brain. Pacinian corpuscles detect vibration. They're particularly good at detecting rapid changes in pressure. That's why we say they can detect vibration. In contrast, Merkel's discs, which we'll talk about in a minute, are very good at detecting sustained pressure. What the Pacinian corpuscles can detect is changes in pressure, and that's vibration. So like when your cell phone vibrates, it's Pacinian corpuscles that pick something like that up because they can detect those changes in pressure. They're located in the deep skin, also in the joints and the ligaments. They look something like what's shown on the slide here. It's an egg-shaped structure, and it has this shape because there's a free nerve ending surrounded by layers of tissue. And they get deformed by pressure, and this is what leads to nerve stimulation. And as we talked about a couple of slides ago, vibration is sent back to the brain via those largest A alpha large myelinated fibers. And then our final structure that we'll discuss are the Merkel's discs. These can detect pressure and also position. They're found in lots of locations, but especially they're prominent in hair follicles. They send signals back to the brain via A alpha large myelinated fibers. And what they're very good at is detecting sustained response to pressure. They're called slowly adapting. And this means that they basically provide continuous information on pressure going back to the brain. And you should contrast this with what we just talked about in the last slide for Meissner's corpuscles and also Pacinian corpuscles. These types of sensory structures are called rapidly adapting because they respond mostly to changes. And that concludes our module on cells of the nervous system.